I want you to think about this for a second. Just maybe 5 years ago, if you brought up designer babies or, editing human DNA,
Yeah.
it sounded like something straight out of a really bad sci-fi movie, right?
No, absolutely. Pure fiction territory for most people.
But today, it's actually changing how medicine is practiced, fundamentally.
Hmm.
We're talking about genetic engineering. And the big news, the thing that really anchors this conversation, is the FDA approving the world's first CRISPR gene therapy. It's called Casgevy.
Approved back in December 2023.
Exactly. So this isn't science fiction anymore. I mean, it's here. And get this, it's now the single most expensive treatment in human history.
And that approval, it really marks a huge turning point. We've moved way beyond just treating symptoms or, managing diseases. We are now literally in the business of rewriting the basic code of life, the inheritable code. So our mission today really is to dive into that, to look at the incredible clinical reality that's happening right now versus the intense ethical storm that this kind of power kicks up. And by the end of this deep dive, you'll grasp something pretty important, these subtle differences in the tech being used right now that, honestly, 99% people just don't know about.
Okay, let's unpack this then. We've got a whole stack of material here. Clinical trial results, bioethics papers, mostly from the last couple of years, 2023 to 2025. And the core question, the challenge, it's massive. How far do we actually push this? I mean, the power to edit genes is already proven. It's here.
Well, the best place to start is probably Casgevy itself, because it's the cure, in action. This therapy, it's designed specifically for sickle cell disease. What it does is edit bone marrow stem cells, basically rewriting their instructions so they produce healthy red blood cells instead of the sickle-shaped ones.
Okay.
And just to make it really clear how real this is, as of, mid-2025, over 115 patients have already started treatment.
115 people.
Yeah.
That's tangible.
It is. And it's active across 50 different treatment sites. We're talking North America, Europe, the Middle East. This is global change happening right now, today.
That progress is just incredible. But what's also fascinating, maybe even a bit startling, is how fast the tool itself, CRISPR, is evolving. I think most of our listeners probably know the basics CRISPR uses that Cas9 protein, like molecular scissors, right? And there's a guide RNA to find the spot.
That's the core idea, yes.
But the original way it worked, it made a double-strand break in the DNA, which, well, sounds risky,
Yeah.
because the cell might not repair that break perfectly.
Exactly right. That initial CRISPR, you could think of it as, a very efficient but maybe kind of blunt cut and paste tool. What's really fascinating now is just how much safer the technology has become, and seemingly overnight. Take prime editing, for instance. That saw its first use in a human patient just last year in 2025.
Prime editing. Okay, so what's different? What does it do that the original couldn't?
So instead of cutting both strands, doing that full cut and paste, prime editing is more like a find and replace function, like in a word processor.
Okay. Much more precise.
Way more precise. It lets researchers make these highly specific single-letter changes in the DNA sequence without making that risky double-strand break. It's just vastly cleaner. And that obviously speeds up getting the stuff from the lab bench to the patient's bedside, radically accelerates it.
And the safety improvements don't even stop there, do they? There was that MIT breakthrough, too. They found a way to essentially turn the CRISPR machinery off once its job is done.
Yes, and that's critical. Absolutely critical. Because the biggest worry, I think scientists call off-target effects.
Meaning the scissors cut somewhere they weren't supposed to?
Precisely. Cutting the wrong gene sequence somewhere else in the genome, that could be dangerous. So being able to just flip an off switch, that's projected to cut down those dangerous off-target effects by potentially up to 40%.
40%? That's huge.
These aren't small steps. They're massive jumps in safety and precision, and you see it reflected in the numbers. When you hear there are over 250 gene editing trials being monitored worldwide right now in 2025, you realize this revolution isn't on the horizon. It's already washing over us.
So, okay, we celebrate Casgevy helping patients, these incredible leaps in precision. But the very fact that these powerful molecular scissors exist forces us immediately to face that core ethical issue, the whole designer-human reality.
The big questions.
Yeah, and here's where it gets really interesting, because you absolutely have to talk about the scientist who broke the biggest rule, Jian Kui.
The scandal.
Right. He stunned everyone back in November 2018, announced the birth of the first gene-edited babies, twin girls, Lulu and Nana. The edit was meant to give them HIV resistance.
The backlash was immediate and fierce. And Jian Kui, he paid a heavy price. 3 years in prison, a huge fine, basically, for illegal medical practice. But here's the unsettling truth. The thing that still echoes in labs and ethics committees everywhere, controversial as it was, unethical as it was deemed, the technology worked.
It worked. The kids, Lulu and Nana, they're school age now, and they appear healthy.
And there was a third gene-edited baby born in 2019 as well.
So the genie is well and truly out of the bottle. We can't pretend we don't have the power now to make changes that are passed down through generations. Heritable changes.
And that success throws us right up against the ethical line that most regulators have drawn, the line between somatic and germline editing.
Okay, explain that difference again. It's crucial.
Right. Somatic editing, like Casgevy for sickle cell, it only affects the person being treated. The changes are not inherited. They stop with that individual. Germline editing, which is what He Jiankui did, creates changes in sperm, eggs, or embryos. Those changes are heritable. They get passed down to all future generations.
And that's the type that's banned pretty much everywhere for making babies, right? Including the U.S.
Correct. Banned for reproductive use in most countries.
But is that consensus holding firm? Because you hear some prominent voices starting to question the ban, people like Dr. Shukrat Metalipov.
Absolutely. The consensus is definitely being tested. Medalipov and others, if we connect this to the bigger picture, they ask a really challenging question. They say, look, if we have proven technology that could wipe out a devastating disease, let's say something like cystic fibrosis, eliminate it from a family line forever, is it actually morally wrong to withhold that power just because we have philosophical feels about what might happen down the road?
That's a powerful argument.
They argue there's huge potential here to permanently end certain kinds of genetic suffering. A moral imperative, even.
Okay, but let me play devil's advocate here for a second. Isn't it incredibly naive to think we can keep a clear, bright line between therapy and enhancement?
That's the million-dollar question, isn't it?
I mean, if the tech works, and if you've got the money, how do you realistically stop someone, say parents, from paying for the gene edit that prevents a genetic risk for heart disease?
Yeah.
Okay, maybe that's therapy. But then what about nudging it just a bit further, improving immune function, or down the line, maybe even boosting intelligence? These aren't just thought experiments anymore, are they? These are actual decisions that we're likely going to face within, what, the next decade?
Absolutely. This raises that crucial question. We're not just talking about fixing obvious diseases. We are definitely entering this very gray area where preventative health starts to blend into enhancement. And that brings us straight to arguably the most troubling part of this whole field.
Right.
Equity, access.
The price tag. It's the ultimate barrier, isn't it?
Yeah.
We mentioned Casgevy, over $2 million per patient.
$2 million.
That's life-changing money. Enough to buy a house, multiple houses in some places. And yeah, insurance companies are starting to wrestle with coverage, but the immediate reality is this technology creates the ultimate haves and have-nots scenario. These incredible genetic cures are, at least initially, mainly accessible to the super wealthy.
And it gets worse. This access disparity, it's actually compounded by a really fundamental bias in the research itself. The data is pretty stark. Only about, 4% of people participating in these big genome-wide studies are of African, Hispanic, or indigenous ancestry. Just 4%.
Okay, wait. Unpack that for me. How does a lack of diversity in the research actually make the therapy less effective for those groups? What's the mechanism there?
Okay, so think about the guide RNA in the CRISPR system. It's designed to find and target a very specific sequence of DNA letters.
Got it. Like a genetic address.
Exactly. Now, if the research data used to design that guide RNA comes almost entirely from one population group, like people of European ancestry, then the guide will be optimized for the common genetic variations found in that group. But other populations have different common genetic variations. Scientists call it genomic variants. If those populations are underrepresented in the research, the guide RNA might not find its target sequels as efficiently in them, or even worse, it might latch onto a slightly different sequence that looks similar but isn't the target.
Creating an off-target effect.
Precisely, a dangerous off-target cut. So the lack of diversity in the research literally risks making the therapy less effective or potentially even unsafe for the very populations who often carry the highest burden of certain genetic diseases.
Wow. So Dr. Andrew Subica at UC Riverside, he warns about this, saying that unless we actively involve minority communities in the development, CRISPR could actually make existing health disparities even worse.
It's a serious risk.
And critics, understandably, worry that this whole trajectory, especially once you start talking enhancement, it could become a sort of eugenics in disguise.
That phrase does get used.
creating discrimination based on who has the better genes or who can afford them.
It's a valid concern and one that needs constant vigilance. Yet, bioethicists are really caught in a tough spot here. They also argue, quite strongly, that we can't let these fears about future enhancement completely stop us from alleviating real human suffering today.
The moral obligation argument again.
Right. If a cure exists, a proven life-saving therapy, is it ethical to withhold it because of hypothetical worries about future misuse? Many would say no. The immediate challenge maybe isn't the technology itself so much as figuring out the governance, the rules, and ensuring equitable distribution. How do we make it fair?
So, okay,
Mm.
where does all this leave us? What does the near future actually look like for genetic engineering? The market predictions are pretty clear.
Oh, yeah. The gene editing industry, it's projected to hit something like $36 billion by 2027. That's serious money flowing in.
$36 billion.
And all that capital, it's just fueling incredible speed. Within the next decade, I think we can realistically expect to see genetic treatments becoming available for things like inherited blindness, muscular dystrophy, cystic fibrosis.
Familiar, devastating diseases.
Exactly. But here's the really critical point. The sort of hidden bonus insight, maybe? Researchers are now predicting that by the 2030s. So not far off at all.
The 2030s. That's practically tomorrow.
It feels like it, doesn't it? They predict we could see the first socially accepted uses of germline editing, the inheritable kind.
Socially accepted germline editing, how would that even start?
Well, it would almost certainly begin with trying to eliminate the most severe, truly debilitating single gene disorders, things everyone agrees are tragic. But once that door is cracked open, even for the best reasons, the absolute need for strong ethical rules and, crucially, global governance structures that can actually keep up with the pace of the science, it becomes paramount, urgent.
So what does this all mean? It really feels like we're standing at a massive crossroads, doesn't it?
It absolutely does.
On one path, we have this incredible power, the potential to eliminate horrible genetic diseases from human experience forever. But on the other path, that very same power, right now, seems tightly linked to the risk of creating totally unprecedented inheritable inequality, discrimination based literally on your DNA.
Mm-hmm. And the choices we make about what conditions we decide to edit, how we distribute these incredibly expensive therapies, and fundamentally for whom they are available, these choices demand broad public discussion. It can't just be scientists in labs or politicians in committees deciding the future of the human genome. It affects everyone.
It really does.
And, the decisions we collectively make, probably over the next 5, maybe 10 years, they could well determine whether genetic engineering is remembered as humanity's greatest triumph or perhaps its greatest moral failure.
The choice ultimately is ours. Wow. And if you think genetic engineering raises some tough questions, just wait until our next deep dive. We'll be analyzing the sudden, massive resurgence of global nuclear energy. Is it finally the climate savior we need, or is it setting us up for the ultimate catastrophic risk? You definitely don't want to miss that one.